The 2020 AACR annual meeting for the first time was held as an online virtual meeting. I had the privilege to present the results of the olaparib durvalumab arm of the I-SPY2 trial in the plenary session, and subsequently there was a special section on clinical trials in breast cancer, which included some important updates on the overall survival results of the EMBRACA trial, which was for germline BRCA mutant metastatic breast cancer. I-SPY2 is a multi-arm adaptive randomized trial platform that runs multiple different arms at the same time. The purpose of the durvalumab olaparib arm was to see whether we could improve pathological complete response rates in a HER2-negative early stage breast cancer population. The trial an arm can graduate for failure or for success. As data accumulates, a statistical program updates the predicted probability of success or failure, and a study graduates when it reaches a particular success or failure endpoint. So the durvalumab olaparib arm graduated with success, implying that it's statistically much more likely to be superior to the control arm than to be equal with it. The final efficacy results showed that the predicted pathological complete response rate improved from 27% to 47% in the triple negative group, and also from 14% to 28% in the ER positive HER2 negative cohort. So these are exciting results because they add to the growing body of evidence that in triple negative disease, immune checkpoint inhibitors indeed improve pathological complete response. And more excitingly, this is now the second time that the I-SPY trial group demonstrated that we can also improve pathological complete response rate in the ER positive subset. We have seen this before with pembrolizumab, and now we see it again with durvalumab and olaparib. What's particularly exciting is that we also identified the subgroup within the ER positive population that we think benefits the most from this triple combination. And that subgroup is the MammaPrint ultra high group that happens to be the upper half of the MammaPrint risk distribution. So these are the patients for whom adjuvant endocrine therapy is the least likely to improve the outcome, and at the same time chemotherapy helps them more. So now we have a way to improve the efficacy of the chemotherapy. So in terms of adverse events, there were no unexpected adverse events. But of course, when three drugs are combined together, the side effects are always a little more than just using one drug, paclitaxel. So the grade 3/4 adverse event rate was higher, 58% compared to around 53% in the control arm. And there were about 19% of the patients who had some immune-related adverse events, grade 3 adverse events. And these adverse events included colitis and thyroid function abnormalities, as well as adrenal insufficiency. So these findings reaffirm the value of immune checkpoint inhibitors in triple negative disease. So it's very clear now from multiple randomized trials that the pathological complete response rate can be improved by including an immune checkpoint inhibitor. The I-SPY2 trial cannot really tease out the contribution of olaparib to this improvement. We are going to do additional biomarker studies around germline BRCA mutation or mutations in DNA repair genes to try to tease out whether there is a population where it's very clear that olaparib is likely to contribute to the outcome. But more importantly, this really pushes the interest further to develop these drugs in the ER positive high risk subset. There are several large randomized trials that have built on these results and explore these drugs now in the ER positive space. EMBRACA was a large randomized phase 3 trial for germline BRCA mutant breast cancers, including about half of the patients who were ER positive and another half who were triple negative. So the primary study endpoint was progression-free survival using single-agent talazoparib compared to physician choice chemotherapy, which included multiple commonly used agents in the second to third line treatment setting. So all of these patients received at least one prior lines of therapy, but some of them received up to 3 prior chemotherapies. So the primary endpoint was improvement in progression-free survival that the trial has met, and it has been published. At this AACR virtual meeting, the overall survival results were presented. And unfortunately, the overall survival results did not show any improvement or difference between the 2 arms. So the investigators very nicely explained that why this is not so unexpected. And the primary reason is that many of the patients subsequently crossed over to receive PARP inhibitor after the progression on the EMBRACA trial. And many of them also received highly effective subsequent therapies, such as platinum therapies. So these results are very much in line and exactly the same as was seen with olaparib in another clinical trial, which also showed an improvement in progression-free survival, but no improvement in overall survival in a very similar patient population with a different kind of PARP inhibitor. I'm not sure that these results really could be interpreted as no survival improvement. It's more like that I think the survival improvement was not detected in the trial because of the complexities of showing an improvement in survival when patients cross over to the same drug after progression. There was a companion presentation that asked the question whether patients who actually have a germline BRCA mutation and also lose in the cancer the normal allele. So each gene has 2 alleles, and patients with a germline BRCA mutation has a defect in one of the alleles, but their other allele is normal. The question was whether if you lose the second normal allele, so both of the genes are knocked out in the tumor, will they actually benefit more from a PARP inhibitor? And the results, somewhat counterintuitively, were negative. So whether you had this loss-of-heterozygosity, allele-specific loss of heterozygosity in the tumor, or you didn't really change the outcome. So the bottom line that we really don't have a good handle what additional biomarker would be beneficial to select patients for talazoparib or olaparib therapy beyond the germline mutation status.